Hello, everyone, and welcome to today's webinar. This webinar is hosted by Jen and sponsored by Asimov. Today's topic is the future of biomanufacturing, AI-driven cell line development and bioprocess design. Thanks so much for being here. I'm Uduak Thomas, Senior Editor for the Educational Content Services Group at Marianne Liebert Publications, a Sage company, and it's my pleasure to be your host for today's webinar. Let me introduce today's speaker. You'll be hearing from Thibault Laurent, Director for Strategic Alliance at Asimov. We'll get to his presentation shortly, but before we do that, let me give you some background on today's topic. Traditional approaches to biologics production, relying on one-size-fits-all expression vectors and empirical process optimization, have resulted in inefficiencies in expression, scalability, and manufacturability. While incremental improvements in cell line engineering and bioprocess modeling have driven progress, the rapid evolution of AI and digital tools is poised to reshape biopharmaceutical process development and manufacturing. So in this webinar, our speaker will share a data-driven vision for the future of biologics production and discuss progress towards holistic, optimal design of cell lines, genetic systems, and bioprocesses. He'll discuss how Kernel, a software design tool that powers Asimov's CHO Edge system, can accelerate timelines and improve titers for cell line and process development. During his presentation, our speaker will cover case studies that demonstrate how this system delivers high-titer clones in less than 4 months for monoclonal and bispecific antibodies. Finally, he'll discuss broader capabilities for integrating multi-omics and bioreactor-scale models to further optimize growth, productivity, and quality. Today's presentation will be followed by a live Q&A. At any point during the presentation, if you have a question, you can enter it in the Ask a Question panel on your screen. All right, let's get started.
Thank you very much for the introduction, and thank you everyone for joining the webinar today. Asimov is applying models much further upstream than traditional biomanufacturing processes. We aim to achieve an holistic optimization of our systems, starting with CHO Edge, our system for the production of biologics. Today, I would like to introduce you to the application of a suite of models developed by Asimov, from genetic designs to bioprocess design, that support better process performance compared to individual standalone optimization. As part of this webinar, I will briefly explain our mission at Asimov. We will go through the tools and models we created around genetic designs and how those apply to cell line and process development. Finally, we will touch about the hybrid and machine learning models developed by Asimov to support our processes. For those who are not familiar with Asimov, our mission is to bring the computer-aided design revolution to biotechnology. In the same way that electrical engineering industry was transformed by CAD tools that brought automation, design rule checks, performance prediction, and design optimization, we are building the first industry-grade computer-aided design platform for our field. The genetic engineering illustration on the slides at the bottom represents how most scientists perform manual genetic designs through simple software like APE, while at Asimov, we are working on our CAD software, Kernel, for part characterization, building genetic designs, assembly, and assimilation. Our first focus is to apply CAD to the design and the manufacturing of advanced therapies. The initial and the most developed application is for the production of biologics, and we will focus on our CHO Edge system today, but we actually launched multiple platforms. We have the antiviral expression platform in 8293, our recently launched AAV system also in 8293, and currently working on our RNA manufacturing platform as well. To give you an idea of how this concept of CAD for biology fits into our platform, we have a suite of hyperfunctional genetic designs and host cells that we can tune using our CAD software, along with our models in order to produce high-performing cell lines to support our clients to reach the clinic. Of course, we use our models and software internally to improve the quality and performance of our services. We have two models to work with our partners, We have the external usage, where we have our systems, our technology are onboarded at the partner site. So the whole system, so Asimov storage system, can be onboarded, as well as, I would say, individual technologies, so like in an unbundled way. And then we have the internal usage through our CS line development services. Moving to the next section, I would like to give you an overview of the tools and models developed by Asimov to support every step of genetic designs. Focusing on our storage system, we develop an end-to-end optimization of biologic production from genetic design to bioprocess performance, including the cell line development part. Starting with our CAD software kernel, which optimizes and automatizes the design of expression vectors based on the molecule to express. This slide contains a screenshot that represents the editor view of Kernel. This is where we visualize and manipulate genetic designs to express the given molecule. The genetic part, called the genetic design, is represented on the top part of the screenshot, and all the parts are individually, I would say, accessible and can be moved through like a drag and drop system. And they can be switched directly with the genetic by accessing the genetic part library in order to, I would say, fine-tune, optimize, or just generate the vector designs of your choice. On the right-hand side, along with the genetic part library, you have access to like standalone genetic tools. So we have, for example, the Kernel AI assistant, we have the Kernel optimizer, and we have the genetic simulator that will predict the RNA polymerase and ribosome flux based on the parts that are assembled within your genetic design. On the left panel, you have access to different views and extra genetic design tools that at the moment are mostly used within Asimov. Asimov with a Troet system is looking to move beyond, or is moving definitely beyond, the one-size-fits-all approach for cell line development by using models to guide vector design. The starting point for everyone is a fixed amino acid sequence. The typical workflow for most CLD platform is to use third-party tools for signal peptides designed for selection, clonal optimization, plasmid assembly. So this workflow is pretty manual, time-consuming, and error-prone. At the bottom, you have the representation of the Asimov approach, where all steps are driven by models developed by Asimov with in silico design and prediction. As I was saying, Asimov is moving beyond the one-size-fits-all approach. So for every molecule we are expressing, we always explore the vector design space. On this representation, we are looking at 6 different vectors that have been used across 4 different molecules and include 3 IgGs and 1 bispecific. You can see that none of the design is giving optimal performance for the 4 molecules. And even in situation, you can kind of like see the opposite effect. Some designs will be optimal for one and will be even the worst design for others. So looking at the vector design space is definitely important to maximize performance and even product quality for your molecule of interest. Taking a genetic design to another level, Asimov has built a genetic compiler that eliminates the manual, error-prone design steps by automating every step of the vector design workflow. So starting with the input sequence, we only need to, I would say, to add the light chain, heavy chain, or each sequence within the genetic compiler, and it will automatically pick the vector design based on the architecture of the molecule, select the signal peptides, as well as perform the clonal optimization and the final assembly in order to finally, as an output, to give you a list of vector designs that should be used to support the expression of your molecule. The different vectors, so the optimized vectors, can be compared to see the different variations, and a genetic simulator can be used to look at the different RNA polymerase and ribosome flux. Taking each step separately, the compiler draws from expression plasmid templates that our cell line development scientists have engineered for optimal antibody expression. It runs simulations using our library of characterized genetic parts and recommends a panel of vector layouts. Simulations are run through the genetic circuit simulator, one of Asimov's foundational models, which is a mechanistic model of expression dynamics that describe the plasmid genetic design, cis and transregulatory effects, transcription and translation fluxes and rates, mass balance of mRNA transcripts, proteins, ligands, and transcription factors. Deployed in Kernel, it really allows scientists to select promoters, UTRs, poly-A, like any part that will help to span a range of heavy chain, light chain expression ratios or glutamine synthetase stringencies in order to explore the vector design space and design, I would say, a set of vectors that will be the best suited to express the, I would say, a particular molecule. Continuing post-translation, we find the role of the signal peptides. As we all know, the signal peptide is critical for efficient secretion of the protein. Some signal peptides are more effective than others at being cleaved and addressing the protein to the right pathway. So we establish a proprietary signal peptide prediction machine learning model that successfully predicts signal peptide type, cleavage sites, and probability. More interested in the model, this is how we used it at Asimov. We were able to mine and identify novel signal peptides that outperform consensus sequences and that we are using routinely as part of our, I would say, genetic designs, and that are accessible directly within Kernel. Then we have the the Kernel optimizer. The Kernel optimizer is an algorithm adapting the codons for a given amino acid sequence to optimize the codon usage, supporting translation, and to minimize problematic motif usage. The Asimov code optimizer, as you can see on the right-hand side, consistently matches or outperforms third-party codon optimizer. The code optimizer here demonstrates is used as part of the storage system for the creation of biologics. However, we see dramatic improvement in other systems, showing that our code optimizer is agnostic to the expression host. So the same code optimizer is supporting our other systems, so for viral vector production. Although it's not shown, the pool titer that you see on the slides definitely scale consistently to clone titers when we reach the final fed batch in number 15. To conclude and with the final step, which is the assembly, the expression vectors designed through Kernel can be directly ordered to our expert DNA team, which will be able to synthesize and assemble the different elements effectively and with all the quality control to deliver high-quality, large-scale DNA preparation that will be ready to transfect. To complete the story and move to the development of stable cell line, our TROET system relies on the Asimov proprietary hyperactive transposase to support the integration of expression vectors into the host and CHO genome. In that case, the compiler directly designs expression vectors that contain the IIDRs to support transposase-mediated integration of the expression vectors. Moving to the development of stable cell lines, the Asimov CAD software works in combination with our multiple host cell lines, with, I would say, our own, including our own proprietary CHO-K1 GS knockout cell line. And our expression vectors and transposase have been validated in multiple CHO host cell lines from different partners, including like, biotech, big pharmas, other CDMOs. And we have all the traceability on the Asimov Choke1 GS knockout cell line. So the original, I would say, the starting bank was coming from ECSCC. The Choke1 line was suspension adapted and knocked out for the GS gene at Asimov. And the GMP bank under ACHQ5 guidelines was generated at Charles River Laboratories. A drug master file was filed with the FDA in 2023. And earlier this year, our own Choke1 GS knockout cell line reached its first IND. Our CHOH service supports the generation of stable cell lines from transfection to clone RCB delivery in under 4 months. As you can see on the timeline, this, I would say, workflow includes final assessment of 46 clones as part in a AMBER 15 14-day fed batch, and include as well full analytical characterization on the material which is being produced. Our labs and headquarters are located in Boston, in the Fenway area, where we use industry-leading equipment and best-in-class practices for every part of cell line development. Thanks to our model-guided choice of genetic parts, we explore the vector design space that allows to identify the optimal design to express your molecule of interest. We have the ability to play with the stringency of the glutamine synthetase transcription units and balance the chain ratios within each vector. As kind of like I described earlier, on this graph, you are not able to see one vector that will provide the best performance for every molecule. In fact, we have a lot of data to support this observation across many molecules, but the tuning of the GS stringency and the chain ratio, heavy chain, light chain ratio is really key, and is really key to support optimal, I would say, expression for every given molecule. Therefore, just to provide an overview, with different maps bispecific that we have expressed at Asimov, we are able to routinely achieve in the range of 8 to 11 grams per liter for clones that have been developed through our cell line development process. Of course, we have definitely molecules that have lower titer, but the aim of this slide is mostly to show that we are definitely able to obtain high performance across, I would say, a range of architectures. I would like to take you through an example. So this is an example that we the molecule we express at Asimov, which is a 3-chain bispecific. So it has 2 different heavy chains with a common light chain. And as we know with bispecific, the titer is not everything. The heterodimer percentage is as important as the titer. So for this molecule, we explore the vector design space, and we look at 14 different vector designs and combinations. We identified optimal conditions that provide, I would say, maximize heterodimer percentage while keeping, I would say, keeping the titer as high as possible. And we selected the 2 pools in red for single cell cloning. And if we were looking at only maximizing titer, we will be missing completely on the heterodimer percentage, which will be like a major issue for the partner. I mean, for Asimov, for the partner as well. So looking at the product and say quality or the formation of the optimal product is as key as the productivity. Taking those 2 pools for single cell printing and going through our, I would say, clone screening and selection workflow, ultimately we were able to generate 12 clones. Here you have the data for 12 clones that have titers in the range of 8 to 11 grams per liter, with the top clones having 11 grams per liter and 84% of heterodimer. Moving to the last section around bioreactor and process modeling. To get from genetic design to process performance, we need to consider the cell population and its overall behavior in larger scale culture. How individual clones have their metabolism supporting and competing with the antibody production. For this, digital biology, so modeling and simulation complement Asimov's synthetic biology and process development capabilities. We are well positioned as a modeling and simulation team at Asimov to pursue a holistic optimal design. Our modeling and simulation capabilities are well supported by our characterized genetic parts, genetic designs, where we optimize the assembly of those parts coupled with multi-omics analysis throughout the whole life cycle between cell line development and process development. So we can modulate in singulation and combination, gene expression, the phenotype, metabolism, bioreactor transport, which represent an end-to-end representation from gene expression to bioprocess. Those models and simulations allows us to predict process performance by optimizing and tailoring the growth, the productivity, and the product quality, so we can be more efficient with our process development. As an application of our multi-mechanistic model, I'm taking the example here of 3 molecules where we identified a titer plateau. So as part of the troubleshooting process, our modeling, cell line development, and process development teams started looking at metabolic network and metabolomics data that we have collected, hypothesizing which shifts in the metabolic fluxes will result in that plateau and how we could eliminate it. So on the graph on the right-hand side, you can see the, I would say, initial platform process, and you, like in purple, you have the model-guided process. So those are experimental data. And the model-guided process was able to result in higher performance, so with a minimum of 23% in titer gain, and up to like more than 100% for molecule 2. With, as you can see, one of the major improvements was around the peak VCD and as well as the maintenance of this high VCD across the 14 days. To get to process performance, we need to look at the cell culture, particularly how cell proliferation scales and competes with antibody production. So for this, we developed our CHO simulator, which is a suite of machine learning, hybrids, and mechanistic model describing cell metabolism, phenotypic states, and bioreactor mass conservation, and bioreactor states. So looking at our CHO hybrid model, as its core, it's composed of a metabolic flux analysis which represents the cell metabolic network, making the assumption that the cells do not accumulate anything, so the mass is conserved within the cells. Our model is trained on experimental data. The flux are modulated by an artificial neural network that we have trained to recapitulate data that we see in various process conditions. So we are presenting 40 dynamic states, 32 for the metabolites and the mass concentrations, 3 cell states, and 5 bioreactor states. So we can obtain trajectory over time of those states and optimize them. So we are presenting here how the model is trained. We have a set of experimental conditions that are represented by the dots, and that represents a wide variety of process parameters, feed strategies, media composition, like different maps. And on the different panel, you can see on the top left the different VCDs. The bottom 4 panels represents different map concentrations, and the other 7 panels represent some metabolites, different metabolites, as well as the pH. So as we train the model to learn these parameters, it begins to recapitulate the experimental data quite faithfully. Now we train the model, we can start applying it. A particular use case here, we predict the trajectory of our baseline process and check the predicted trajectory we check that the predicted trajectory matches the expected baseline. So the predicted baseline is represented here in blue. And then we can run an optimization problem to maximize the endpoint titer, which is represented here by the green curves, the green lines. The titer is represented on the bottom right panel, and by kind of like running this optimization problem, we can provide certain decision variables for this process. So, for example, we are going to leave the variable should be the feed quantity, but the feed timing will stay fixed. And after we can let the model decide on the optimal variable, the optimal, I would say, on the optimal range to work within the variables. So after this is kind of like the model guided process. And when we look at after experimentally validating the model guided process, we were able to identify some major improvements. So for one of the map, we obtained 20% titer increase, while for other maps or multiple cases, we were able to obtain around 7%. Not only we can use a CHO simulator to optimize processes for individual clones and products, we can apply it to improving our CLD workflow. In our typical CHO process, we will screen and advance the top 46 clones based on productivity and monoclonality assessment. So in the Amber 15, as a final selection screening step, the 46 clones will be assessed through just one media and feed system, which is our platform process for the 14-day fed batch. Using the CHO simulator, with the clearly defined objective functions, we are now able to reduce the number of clones advanced to 16. So we are going from 144 clones down to 16 while we are still in very small scale. So the machine learning model is using this kind of like small scale data set in a much more intelligent way. And the idea here is not to, I would say, obtain a ranking of the clones, but more to like ensure that the top 16 clones that are advanced are, I would say, are within the top, are part of the top clones that should be advanced if we had the 46 clones. So reaching the clone selection stage in the Amber 15, and that's part of our platform 2.0. We use now the model selected feeding strategy to screen the 16 clones and to find the optimal combination in terms of feed strategy and clone phenotype. So each feed strategy can be really like amenable to a specific clone and molecule. This model-optimized feed strategies welcomes the molecular and clonal diversity in each CLD campaign. On the left, the platform feeding strategy is represented with a black line. So this is really like the platform process where there is no variations in the feeding. And for each of the feeds, we have feed 1 and feed 2. That could be mentioned as being like cell boost 7A and 7B. And for the model guided process, we have two, I would say, alternative strategies that are called feed strategy A and B, and that are optimized by the model and captured in green and blue. Overall, for the strategy B has a higher cumulative feed compared to strategy A, and strategy A has a lower feed during the exponential phase. So you can see that strategy A, the feeding is starting on day 2, while in feed B, the feeding is starting, for example, on day 1. Looking at the experimental assessment on the right-hand side, For example, if we take the clone 4, which is in red, it shows a negative response to strategy A, but a titer increase of 11.7% in strategy B, while clones, for example, 1 and 3, show a complete opposite response. So for this application, the objective function was to embrace the diversity. So we would look at pretty different strategies, right? So we definitely expect some complementary responses based on the clone. The idea at that stage, and this is the first time we are screening the clones and looking at the clones in a fully controlled environment through a 14-day fed batch. We are not looking to fine-tune the feeding strategy, but to find the optimal, like the baseline feeding strategy to really outline and identify, see maximize the performance of every single clone. So applying those feeds to 5 clones expressing the same molecule, we are able really to demonstrate different responses, leading to between 4 and 38% titer improvement in our fed batch process compared to our baseline platform process. This example concludes the description and the application of our suite of models that we use to drive better performances from genetic designs and all the way to process modeling at Asimov. Okay, so we are getting to the end of this webinar. And to summarize, at Asimov, we developed and are constantly improving our kernel computer-aided design software, our cell lines, genetic designs, even transposase and models to support an holistic way our different expression systems. Thank you very much for listening. I will hand it back to Jen, and definitely I will be happy to take a few questions.
Many thanks to our speaker for an informative and interesting presentation. We're going to begin the Q&A session in just a few moments, so please go ahead and submit your questions if you haven't done so already. Before we start the Q&A, a reminder that this webinar will be available to view on demand in the next 24 to 48 hours. So please share the link with your colleagues who might not have been able to join us today. And a very warm welcome to the live Q&A portion of today's webinar. Thank you so much for being here. Thibault is ready to take your questions. So as a reminder, if you have a question, please go ahead and enter it in the Ask a Question panel located on your screen. We've got lots of time for questions, so please go ahead and get them in. Thibault, once again, thank you so much for that presentation.
Thank you very much.
No, my pleasure. And I'm going to go ahead and get started as I do have some questions that have come in for you. First question I have, what data / measurements do you use for the ML model in CLD?
Yeah, so that's a good question, a pretty broad question depending on the application. So in the presentation, I was covering a couple of examples. So for example, the use of our machine learning model in order to to reduce the number of clones that we are selecting during our, I would say, clones selection expansion process. So in that case, the model will be based mostly on productivity data, on cell growth data, some metabolites as well. And those will be compared as well to, for example, historical data. So how they behave compared to historical data. So that would be really to down select the number of clones. That's a very specific situation. So it's a pretty simple, I would say, model which is based on, I would say, small scale data and pretty limited data sets. However, we are using as well machine learning more for the optimization, or as I was saying, the screening of different feeding strategies, mainly to look at the scope of, I would say, clone phenotypes that we can get, because we are seeing a lot of heterogeneity, and we are seeing that sometimes we are ignoring, or we are forgetting some clones just by using a standard platform process. So here, again, we are going to use the same data, but since we are a fully controlled environment, we will be able to look at other process parameters. And the idea is to look at historical data because we are seeing I would say a lot of data are really like translatable across molecules. So those models are fully trained based on historical data.
Excellent, excellent. And kind of a related question to the first one, also having to do with data. And I think you may have started to answer it. How much data do you need to train the models?
Okay, so that's a broad one as well. So that would depend. That would really depend. So if we start more with the genetic design aspect, so if we speak about the code optimizer and for example, and signal peptide cleavage predictor, those are trained on publicly available data sets. So on our end, it doesn't really require like tuning for specific molecules, host, or other context. For example, after we have the genetic circuit simulator, where we have all our parts being characterized and using combination. So that one is based on first principle model. So it doesn't require any training. So we really as I was just saying, if we measure the attributes of each individual genetic part and in high throughput fashion, so they have been all characterized at Asimov, and we can simulate their performance in combination. But after we have the extreme, which is related to the previous question, where we have more the hybrid process, bioprocess simulation model, and that one is being trained on can be trained on a very small data set, depending on what we are looking to achieve. But it can be trained as well on, let's say, 90 bioprocess runs across different scales, molecules, feeds, temperature, pH strategies. And we have found that those, I would say, observations, models, and they are kind of the output is predictions performance that are really agnostic, for example, to the molecule. So this is pretty good, and that's what we use.
Excellent, excellent. Next question I have: what parts of the vector design space do you explore?
So I briefly touched about this. I would say we work at different level. And that is kind of related to something I mentioned, where we are working from a set, for example, of genetic designs that we I would say pre-engineer or pre-develop. So every time we run the compiler is not going make new design from scratch, because we will be reinventing the wheel every time. So we have a set of pre-established designs, got a very, very large set. We are speaking about dozens and dozens of vectors, and in those vectors, we are if we compare some of them, we will be just switching one element, for example, one promoter, but that shift will have a drastic impact, for example, on the heavy chain light chain ratio within the vector. We are looking as well at, for example, at positioning of the different transcription units, so the GS, heavy chain, light chain. We are looking at, I would say, orientations. We are looking at, of course, shifting some very specific elements within each transcription unit. So there are many levels we can play. Yeah.
Excellent, excellent. And just to remind our audience, if you have a question, you can enter it in the Ask a Question panel on your screen. And thank you to those who've already sent in questions. The next question I have from our audience: do you find your in silico analysis keeps sending you to evaluate the same subset of elements in your expression vector?
Yes, that's exactly what I was saying just before. The idea exactly is not to reinvent the wheel every time. To be fair with you, that's correct. If we work with a standard IgG, IgG1, IgG4, we have some, I would say, a set of vectors, 6 to 10 vectors that are really, really good. And we know that with those designs, we don't need to reinvent those designs, at least not every time, because we know that they can perform really well. I would say, as I mentioned, the paradigm on one size fits all. So we will have at least one design that will perform really well compared to the others. What's happening kind of in the background on our end is that we have our Asimov Labs, which is our foundry team, that does a lot of additional part characterizations and further implementations in our genetic design process. So we might have a set of vectors that we can apply to some specific molecules, but still every few, I would say, we have a lot of iterations happening from time to time. So those designs will change by themselves. Of course, if we start speaking about more complex molecules, yes, then that definitely will have much more variations within the designs. Yeah.
Excellent, excellent. Next question I have, is it possible to access only some elements of your CHO Edge system?
Yes, yes, correct. I think I didn't mention that. Yes, that's a good question. Yeah, that's correct. So we worked with our partners, for example, on a CHO takeout, so more like on the external usage of our system in an unbundled way. So yes, we are happy, for example, to provide our transposase, our vector system, and for you to access within your, for example, CHO-GS knockout cell line. That's something we are used to do, and I think I mentioned that, but our system has been validated already across multiple hosts, from multiple partners. So we are confident that our, I would say, specific technologies within the CHO Edge can be transposed and transferred to your labs and your, I would say, cell line. Yeah.
Excellent. Does the genetic compiler support different molecule architectures? Excuse me.
Yes, that's a good question. Yes, but still as well in development. So to some extent. So at the moment, the genetic compiler can support, I would say, standard IgGs and some, I would say, 3-chain bispecific molecules. But if we start going to like more complex ones like trispecifics, or where we have like more like chain stoichiometry is getting very different, or outside of the normality, then we are working on the V2 version at the moment that will be able to support that, because this is much, much more complex. Something I just want to clarify, or maybe to mention, is as part of the genetic compiler, we are, I would say, able to predict, to give optimal design vector designs based on architecture, not based on sequence. So just wanted to mention that. So from a sequence, we can't predict, which design would give the best titer. This is more on the architectural level. Yeah.
Excellent, excellent. Next question I have is, let me read the full part out. Does your model optimize for multiple objectives like titers, cell viability, doubling time, and product quality, or is it mainly focusing on just the titer?
Yes, technically we can do all of that. So that will depend on which variables or which functions we want to kind of like to look at, or the ones we want to lock and the ones we want to, like we say, to leave open for variation. So that would depends. I would say across our process, across our platform and all the the campaigns we have achieved and process development we have done. Cell viability is not something that we had to optimize a lot. We usually retain a very high viability through our 14-day fed batch. So what I mean high, meaning more than 95% viability. So this is not something that we need to work on. However, product quality, yes, is pretty key. So by product quality, if we look at PTMs, so on the glycans, on the charge variants, yes, that's something we have been working on quite a bit. Many by in collaboration with some of our biosimilar biosimilar partners. So yes, that's something definitely we are doing. So no, titer is not all. Definitely it's a key thing for everyone, but this is not all. Yeah.
Excellent, excellent. And the next question I have, how is the screening process done in 4 months from transfection to clone RCB preparation?
So screening process, I will assume clone screening expansion process, selection process. I assume, but maybe I'm thinking about, I'm going to cover a couple of things. So, so when speaking about screening, they are thinking about pool and clone. So because ultimately, like at first, we don't start from a single design, we start with a set of design. Therefore, we are, I would say, starting with, I would say, a good number of stable pools that will be assessed through, like, for example, like batch or fed batch. And therefore, we will be looking at productivity and product quality. And then from there, we are screening the I would say, the best pools, looking at all those data and selecting the top ones to move to single cell printing. That step is done in, like, I would say, pretty quickly. So we have, I would say, a very high throughput at Asimov at different stages, so analytical development, cell line development, and as well as, I would say, as part of our DNA team for generating all those designs, the vector designs. So we are not losing any time because of this kind of like increase of samples that we need to look at. At clone stage, clone stage, I mean, we go through like a pretty standard process, which is, we are using the F-sites and the symmetric instruments for single-cell printing and I would say growth tracking and monoclonality assessment. But after we have two different steps in very small scale, so 24, for example, deep, like in 24 deep well plate, where we are able to very early on select and exclude or select, I would say, the clones that we are going to move forward for the Amber 15 for the ultimate 14-day fed batch. Our cell line has pretty good doubling time, so that allows us as well to stay within that kind of like 4 months window. Of course, these 4 months can vary a bit depending on the complexity of the molecules. That can vary a bit, but that's what we are aiming to achieve.
Wonderful, wonderful. Thank you so much, Thibault, for once again for your presentation and for taking time to answer some questions for us. I really appreciate you. And to our audience, that is all the time we have for today's webinar. Thank you so much for being here and for asking such great questions. Before we go, a quick reminder that this webinar will be available to view on demand in the next 24 to 48 hours. So you should get a link in your email, and we encourage you to share the link with your colleagues who might not have been able to join us today. So once again, many thanks to Thibault for being here and for such a great presentation. I'd love to thank today's sponsor, Asimov, for sponsoring this event. And I'd also like to thank the Gen Multimedia Group for all of their support. So thank you, David Mosley and Reynolds Gutierrez. And most of all, thank you to you, our audience, for being here. For everyone at Gen, I'm Uduak Thomas. Have a lovely day.